But it's an honor and privilege to be here, and I'm really impressed, more than impressed, and the vision that CHI has. We have developed this NCCC grant has led the way for Center for Translation Research, and it is amazing that CHI has the vision to establish such a project. And what I will try to do when Jeff asked me, I want to give an overview of what's translational research and how it applies into the clinical practice that we have. So as everybody knows, the cancer is a genetic disease, and it's a disease of the genome. As the prior speaker mentioned, every single cancer cell is different. Every cancer patient is different. Every tumor is different, and every tumor cell, as time goes, becomes different. So it's a very heterogeneous setup. And the goal is to cure cancer, which has eluded researchers for so many years. And the cancer lifespan, we want to extend that, narrow that gap, and then make people live. And again, as heterogeneous as it is, cancer starts as an initiation, malignant transformation, and then becomes lethal type. And translational research, if you look at the literature, the earliest mention of it goes to 1993, but it was not in the sense that we are using today. Until 2005, when Dr. Zerhoni, the director of the National Institutes of Health back then, came with this paper in the New England Journal of Medicine after NCI and NIH decided to come with a new roadmap and then new project, and they announced the CTSA, Clinical Translational Research Award, and they funded institutions for translational research, and so far I believe they have spent $500 million in that to support academic and private institutions. Translational research in Italian, there is this proverb, and I looked up the Italian translation, traduttore traditore. It's loosely translated as to translate is to betray, and then it refers to interpretation of the literature, and you have to interpret the literature in its own language, and there are subtleties, intricacies, and the other parts that can be lost in translation. And this expression, it implies to that translator, traitor, and they are very similar. So in the translation of basic science into the clinical science has been problematic, and that has been very slow. It's a paradigm of research alternative to the dichotomy of basic and research and the clinical research. It aims to inform the clinic and the laboratory with the results of each other's work. Traditionally and historically, they have been not communicating well. And in a diagram, it's a scientific process, bench to bedside, and then back bedside to bench. And the basic science requires a knowledge-driven ecosystem in which the constituents generate, contribute, manage, and analyze data available from all parts of the landscape. So some researchers defined it as what occurs from bench to bedside in the development of new drugs, devices, while others view it as a translating research into practice. So there are two views, the product-driven or community and policy focused. And then always it has been bench to bedside, but actually it is bench to bedside and then also bedside to the bench. It's a complex dance from back and forth, as shown on this diagram. So it starts in the basic laboratory bench side, and then NIH, when it came up with the CTSA award system, they described it as a two systems, the T1 and T2. And I had the wonderful opportunity being at the NCI. The first year as a clinical, but my last 2 years were basically working in a lab in the bench like a PhD. So I had the luxury and the advantage and privilege of seeing both sides. So when NCI described it as a T1 and T2, basically the discovery in the bench in the basic science research on animal models, and those are our animal friends. We can thank them enough as the mouse models and the other guinea pig models that provide us a tremendous opportunity to understand the biosystems. So that biologic bench research translated into the bedside, which in our clinical practice, phase 1 and phase 2 trials, they are tested. So NCI, when first they announced the CTSA award, pretty much they focused on T1 and T2. And then notoriously, the research money has been by NCI mostly on the T1, and beyond the T2 has been not as much. But later on, several authors came and expanded this T1 and T2 concept from T2 to T3, T4. Basically, they said the original definition, 2 labs, the bench and the bedside, the clinical practice, but there is more of it. There is the guideline development, there is meta-analysis, there is post-approval outcome research, and then implementation, cost analysis, comparative effectiveness. That includes all of them, and then the spectrum has expanded from T0, where you have the concept and idea, and then it goes into the first human phase 1, phase 2 trials, and it goes to phase 2 clinical trials, which they put it as T2, T3, and then dissemination to then T4, public health, prevention, population health impact, then T5, social health care practice, security, economic opportunities. So as of today, the most extensive and explanation of has been T0 to T5. And what is really important here to emphasize is the delay. Why NCI came up with that 2005 CTSA award was that the delay and the resentment from the public that all this basic science discoveries does not translate into clinical practice and doesn't help the people where they desperately need a cure. So there has been a definition of value of death, which is here. So the discovery, target validation, lead optimization process, there is this part that there is a huge gap, and then it takes 14 to 17 years for 14% of basic science discoveries to get into the practice. At average, it takes 17 years. And that has been due to several blocks, and it can be at the T1 level, at T2 level. Some of them are listed here. At T1 level, the lack of willingness to participate, regulatory burden, fragmented infrastructure, and then incompatible database, lack of investigators. That's another topic going on in the medical schools now. The curriculum includes the translational science. The emphasis in the old days when 1945 National Science Foundation described the basic science as a science to understand the biology and the mechanisms of biologic systems, but not necessarily must have a clinical outcome applicable to clinics. So with that definition, even my fellowship years, I remember we had the luxury of choosing a lab that we would like, and we would go and talk to the lab chiefs and mostly basic scientists what's their project all about and if we would join them or not for our next 2 years of basic science or clinical science. The concept was there that they were creating data without the force or urge. Is this data would be applicable to clinical practice the next day, the next year, the next 10 years. They were just trying to understand. When I asked one of my attendings I was interviewing, he was working on differentiation of cells, and naively, I was an MD background. I happened to ask, so can we use this cell culture information to apply to our leukemia patients? And I saw his face kind of turned, and this is the MD thinking. He said, you don't jump into the clinic, you have to mature the data, understand it, which was correct in a way, but that luxury and urgency now is pushed by that you have to come up with clinical implications because we cannot afford sitting 17 years to get a basic science discovery to clinical use. So in a way, this translational science push coming from that they want to reduce that value of that and get the stuff into clinics. And then traditionally, historically, NCI money has been mostly spent on T0, T1 projects. Beyond T2 was only 1.8. Lately now we can see that 30% of the budget is going to clinical research, but still it's not good enough. So one of my patients, he's an advocate, as a physician himself, myeloma advocate, and he's a survivor. He said, Sidki, make sure you read this, and I will read it. It's nothing. A few stem cells, another 75 years of research will or can fix it. So to emphasize that there is that lag. Again, back to my, back then our director was Dr. Harold Wormus. He's a Nobel laureate, as you may know, discovered oncogenes. He has this concept, clinical science and the basic science, two different words, different languages as it comes to the translator, traditore, cultures, goals, tools, personnel. And they were like that until 1990. Again, if you look at the literature, there was SPORE program, the Special Projects of Research Excellence, and pancreatic cancer, and at the University of Nebraska I'm affiliated with, had a great deal of success with that. That was pushing the basic science and the discoveries into clinical practice. And Harold Wormus always has the basic science concept, and he came up with this his second term, became the NCI director, provocative ideas, that you want to have some provocative thinking in an open freedom for imagination and the science, but yet apply it to the clinical practice, just don't leave it as a concept. So after 1990, when translation science become more and more visible or understandable, now the clinical and basic science kind of merging. Hopefully MDs and PhDs are joining forces, and it hopefully will not be two different cultures. It will be one joined term and then practice, and that way it was all our envy at the NCI. Again, as a fellow, we would love to be an MD, PhD, and some of them were MD only, some of them were PhD only, and we would admire the ones MD, PhD. But it should be all together overlapping now with the way the science and the things are changing. So I will try to give you some fascinating success stories, how translational science become applicable to the clinical practice. And again, we are one of the fortunate generations that we witnessed. We lived through it. As I think about it, we lived through all these discoveries, even though sometimes we take it for granted. In the old days, it was just the clinical or versus basic science. But here, you can see this rituximab story, trastuzumab story, 97 through 2011, last 15 years. There has been very fascinating discoveries. So one of them is the rituximab story, which is rituximab is a monoclonal antibody. It binds to the CD20 on the cells of the on the surface of the B cells. And the trick here that the scientists exploited was CD20 is present on all B cells, but it is not present on maturing or young B cells. So it's a selected target, and they came up with this antibody which attacks to CD20 and then triggers the body's immune system. And then it creates a big advantage in overcoming this lymphoma. So again, I want to emphasize, it started in 1988. A protein CD20 was identified. This is pure basic science. So if you left it there, did not take advantage of it, it would be basic science. And then they said, okay, could it be a target? Let's develop a monoclonal antibody. Again, pure basic science. Monoclonal antibody was developed. 1993, and look at the time distance, not too bad in this case, 88 through 93, a monoclonal antibody was developed, and then first study in 1996, so 8 years there. And then approval was granted by the US FDA, 97, so 88, 97, 9 years. This is the fastest, I think, that as good as the success of that rituximab is. And then what makes rituximab also significant is the first therapeutic monoclonal antibody approved for this treatment, and also first single agent approved for therapy of lymphoma. And here is the lag time was 9 years. And then if you look at how from the basic lab bench to the bedside, it made a huge impact. The non-Hodgkin's lymphoma mortality rates from 85, they were on the rise, and then once rituximab approved, they're coming down, and there are other drugs, other generation of monoclonal antibodies recently being approved one after another, making it even more refined and perfect. So this was a great success story. Another great fascinating success story is HER2. Even I was in internal medicine, I have heard about HER2, and our attendings in oncology, I used to take my electives rotations because of my interest, has mentioned me, or you will be working on HER2, and it is 1989, 92, and it was there. We all knew about HER2, but it was not until 2005. And what is the history on that? It was first identified as an oncogene activated via point mutation, chemically induced rat neuroblastoma that HER2 knew comes from neuroblastoma. And then it was found to be overexpressed in 20 to 25% of breast cancer cells. In 1980s, a monoclonal antibody against HER2 was developed. And I think that rituximab story helped this too, the monoclonal antibody. They saw the example that, yeah, it is working. Maybe we should develop monoclonal antibodies to other targets. So this was 1998, first time it was used on the metastatic breast cancer cells. Again, this is happening while I'm practicing too. I started at St. Francis 1995, and I had a couple patients that metastatic recurrent refractory breast that I put them on this HER2 trial. Then there was a lottery, actually. They were pulling the lottery. If patient wins it, they would be on Herceptin, and one of my patients lived 10 years thanks to that protocol. But back then, we didn't know what it was like for everybody, or would they be checking for HER2 positivity or not. What is fascinating about HER2 and the trastuzumab story is that it's not only first monoclonal antibody targeting an oncogene, not a cell receptor or anything, but the oncogene, basically. And then it was also launched with a companion diagnostic. So it turned out that if you give it to people with the breast cancer who don't express the HER2, they do not respond at all, and it has to be a biomarker driven. So that was a turning point for Herceptin story that showed in real life that this is something that we should be doing. And the most fascinating stories of all is the imatinib story. Again, it will give you the point. 1960, Philadelphia chromosome was described in Philadelphia. Peter Novell from University of Pennsylvania and David Hungerford from Fox Chase, they were both in Pennsylvania. So they saw under the microscope 922 translocation as Philadelphia chromosome. And it took how many years? 2001. It took 40 years that Philadelphia chromosome discovery became such a practical, wonderful, magic bullet home run. It's 40 years. How did it happen? What happened in between? 1973. Janet Rowley identified the mechanism by which the Philadelphia chromosome becomes through a translocation of two chromosomes. 1983, Heisterkamp et al. Demonstrated human CABL oncogene is located in the region of chromosome 9 that translocates to 22, comes together with the BCR breakpoint cluster region. And then 1990, Daly et al. Showed that if they could induce this mutated oncogene into the bone marrow cells, they created a model like chronic myelogenous leukemia. And then 1990s, they identified the function of BCR-ABL. In 1996, again, there's fascinating stories. In the interest of time, I'm going too fast, but drug companies, they did not see a big catch on CML. There are 48,000 cases a year, I believe, total. So if you're a drug company, you want to invest your money with the most common cancers, lung, breast, colon, all that, 4,800 a year, who cares? And very fascinating. And then Novartis company kind of did it out of altruistic purposes. Let's say, let's do it. Brian Drucker. Drucker was working on it, and they got this small molecule to block that tyrosine kinase pocket. And their goal was just to create some model, but honestly, the company itself even didn't think they would make money out of it. And 1996, STI-571, STI stands for signal transduction inhibitor, was tested in cell cultures. And Brian Drucker elegantly expressed his experience with this, that he was giving this drug at the lower doses, phase 1, and nothing was happening. They increased it, and nothing was. Later on, they see some hematologic response, blood counts were getting. Patients had no side effects, and they kept giving it, and they would see that, amazingly, the blood counts were normalized. And a small phase 1 trial, 31 patients showed complete response. In a phase 1 trial, if you get 20% response, you jump. You are excited. Here, 100% response, not to mention no side effects at all. So then it was discovered that this was the magic bullet because the CML is driven by just one oncogene. That's what makes it so great discovery. Unfortunately, not all cancers like driven by one oncogene. So it was just perfect solution to the problem that they put that molecule imatinib, STI-571, matches that place where the ATP should be binding, and then it shuts down all the cascade of events beyond that stop. And even we don't have to know basic molecular science, what is beyond that step, but all you need to just shut it down there, then you almost cure the CML. And when STI come again, 1999 or 2000 ASCO, even the big experts in CML, they were looking at it, yeah, this might be stopping the transplants. Until then, all the transplants were the only cure, which with big high 15% mortality. So they even made the analogy that STI stands for stop transplants immediately. In one of the talks, Drucker said, no, it is not stop the transplants immediately, select transplants intelligently. And then still it has a value, but the imatinib has done a huge impact on, and it became the model magic bullet. Then there's another fascinating story, anti-angiogenesis story. Again, fascinating story. Judy Folkman, one of my, I admire him for his vision that he was a surgeon, but he was a real amazing thinker about this, described it back in 1971. Angiogenesis and very elegant experiments in the cornea of the rabbits. Amazing. And yet, it didn't get into action until 1989. VEGF was identified, and then VEGF stands for vascular endothelial growth factor. Then clinical trials didn't start until 1997. Initial endostudin was a failure, but later on, they discovered the monoclonal antibody, again, connected from rituxan or Herceptin, and monoclonal antibody against vascular endothelial growth factor. It showed a great, not great, but a very applaudable success. And again, my patient asked me to make sure to read this. Go home today, they cured me using this new miracle drug. I'm afraid it will be years. So there is this delay, unfortunately, and I cannot thank enough our mouse models and the guinea pig models, and even I make the joke, Heather knows that, we don't get consent from these poor animals, we just put them on the trials, and yet I have to go through a huge regulatory burden paperwork to put the patient on a trial. So anti-angiogenesis story has exploded over the years, and I would say decades, unfortunately. Then they have discovered VEGF A, VEGF B, then receptors 1 and receptor 2, receptor 3, and then since the initial Judy Folkman's description of it, it took how many years? I guess 30, 40 years that we have now approved drugs for indications of lung cancer, colon cancer. Initial one was then breast cancer, kidney, brain cancer. Just yesterday, FDA approved ramucirumab, another VEGF monoclonal antibody for gastric cancer, which is a very difficult cancer to treat, and it will open some new avenues for our patients. So that's another fascinating story, and this list will keep going longer, I expect, with the addition of ramucirumab just yesterday. Then there is this lung cancer story, and again, the money is in the big common cancers, lung, breast, colon, but as a resident and fellow, when they will ask me, lung cancer, small cell, non-small cell, and then non-small cell can be only 4 types, squamous, large, adeno, that's it. And yet, in 1987, they described the KRAS mutation. Then it went on, and 2004, they described the EGFR mutation, and then still a large bulk of it unknown. It's not one single disease, lung cancer. Then the Tom Lynch and his group from Mass General, and then some groups in Sloan, Memorial Sloan, and the Boston, they were giving this gefitinib story, is a very interesting one too, Oresta. They were giving it to patients with lung cancer against the epidermal growth factor receptor, not knowing that how well it will work. And it was working, but some patients was having fascinating remissions, and yet some, it didn't do any good. So that observation also led to who was responding, the non-smokers, Asian, female. Then they decided, let's check what's going on, and when they analyzed their EGFR, there were those mutations, exon 18, 19, that triggered, and all of a sudden that was discovered that, yeah, that drug was working wonderfully on these specific EGFR mutation patients on the subsets. And then that led to more evaluation, and they investigated ALK, anaplastic large cell lymphoma kinase. That's also well known in lymphomas, for anaplastic large cell lymphomas for decades, but never was thought to be in lung cancer. They discovered it, and then there was this phase 1 pill, crizotinib. My friend again, Chris Takimoto in San Antonio, he gave a nice talk on that. They were seeing this fascinating responses when phase 1 trials, as you know, you give all comers, all cancers. Some of these lung cancers were having wonderful responses. They go look back and check what is that these people responding. They found out they had the ALK EML mutation. So here's the best example of that translation research, bench to bedside. Then here's the bedside to the bench. They figured out, oh, EML ALK is responding to crizotinib. So they started working on the testing for EML ALK and then putting them on trials, which led to the approval of crizotinib, even without a phase 3 classical randomized trial fashion. And today, starting 2003, this is when you ask an oncologist or physician internist, it's almost the lung cancer. Now this is lung cancer, and I bet it will get even more complicated. And then EGFR mutations, story started with them, ALK, EML, RAS mutation. It works in some mutation with the lung cancer RAS mutation, and there will be more to come. So they were fascinating examples for treatment. And there is also a huge, as this CTR is all about, too, the continuum of biomarkers. The same way translational research starts from biomarker discovery all the way to clinical practice and goes through the same steps. And what we have benefited from biomarkers in several ways, the prevention, screening, diagnostic tools and then again, their story is decades or years to get to the clinical use. So BCR-A1, BRCA1, and BRCA2, it was originally described in a 1990 paper in Science. By the time the Myriad company got the patent, and that has been a huge discussion too, that can you patent the genes or not, but by the time that became a usable test, there was 6 years. And the similar story on HER2, that a nucleic acid research paper back in 1965 described it, and then by the time it was Dacotest, Herceptin, Dacotest was approved, it was 13 years. And then came up the gene expression panels. There are several of them. This is just one of them, Oncotype DX, there's MammaStrat, MammaPrint. And that took years. And then how do we utilize them? To diagnose, to screen, to make a decision to treat or not to treat. If they are HER2 positive, you make a decision to treat them with the Herceptin. If they are negative, you don't. And then if it is KRAS mutated, again, that's a fascinating story too. This Erbitox came back as the antibody to against the EGFR, and was not working in some patients, was working in some not as good. That turned out to be, if they were KRAS wild type, they were responding. And again, I'm sorry that I'm rushing because of the flight time. Then I want to just touch base a little bit what happened with the Human Genome Project. And I love the way Dr. Bert Vogelstein at Johns Hopkins here has a very good approach to this, a Human Genome Project. Again, 89 through 2000, it was going on, an international project. Just to simplify, each gene is a page containing 15 characters, the way he describes it. Then the genes are organized into books, which are the chromosomes, 1,000 pages. Then genomes is an encyclopedia of 46 books, from 23 from mother, 23 from father. And I find the analogy interesting here. It costs $3 billion, and they approximate that the base pairs are 3 billion base pairs, so it comes to $1 base pair. And what it did was, there was a huge expectation when the Human Genome Project is done. They thought that we will cure Alzheimer's, we will cure cancer, we will find all the problems, what it's causing. And it gave a huge insight, but unfortunately, it didn't accomplish what people were expecting in this genome-wide association studies. It like exploded. By 2011, there were description of 155 disease loci marked by single nucleotide polymorphisms. Yet, it didn't turn into they discovered a whole bunch, Huntington disease, Alzheimer's, diabetes, but it didn't turn into a real cure, and this was mentioned as like drinking from fire hose, so much information coming, but are we able to even utilize that? And New York Times published this 2010, A decade later, when Bill Clinton announced the completion of Human Genome Project, everybody was thinking this is in 10 years we will cure a lot of diseases, but it turns out that both cancer and Alzheimer's has been elusive. We didn't cure them 10 years after even the completion of the Human Genome Project. And then they said, okay, maybe we can go look at if we can sequence all the human genome, why not just sequence all the cancers? So they did come up with the Cancer Genome Atlas Project, TCGA, and they sequenced all the cancer genes. And again, Bert Vogelstein, John Hopkins group did a lot of work on this. A lot of these genes are international, national, but they have a lot of them from Bert's group. 11 colorectal, 11 breast cancer, they were published. They identified these genes, cancer-related genes. And initial thought was oncogenes. Harold Wormus got his Nobel Prize for oncogenes. Oncogenes meaning the driver genes. And turns out that most of these genes are not oncogenes, they are suppressor genes. Just to give what they found on the Cancer Genome Atlas was, there were abnormalities in the cancer, but that wasn't as big as everybody expected. If you make the analogy again to going to encyclopedia of Dr. Vogelstein's, 44 pages with typographical errors that indicates the mutational stuff changes, the base changes. 11 missing pages, that's the deletions. 8 duplicated pages, like insertions. So then again, as Bert puts this very nicely, if you are publishing an encyclopedia of, 46,000 pages, and you have this many typographical errors, it's a great job, actually. So then we are empty-handed again. How are we going to hit the cancer? So the turns out that all these genes, novel cancer genes discovered through the genome-wide sequencing and all that, tumor suppressors, only the orange colors are drivers, the oncogenes, the mutations that drives the cancer. And then unless you have an oncogene, it's a good target to hit so you can stop. But if you have a suppressor gene, how are you going to hit it? It's already suppressed. You have to remove the suppression, but it's not that easy as hitting the oncogene. So what Bert suggests that more than the oncogenes, the tumor suppressor, or other genes, you have to target the pathways. And they have described around 12 pathways. And Bert thinks it's pretty much it, but I'm curious, maybe there are more pathways, what we don't know. But his point is well taken. What he says that curing cancer, instead of targeting the mutations, we need to target the pathways, like PI3 kinase pathway, apoptosis pathway, angiogenesis pathway. And I'm sorry that it doesn't reflect too well, but there are total 12 pathways that we are well aware as of now. And as opposed to finding an oncogene to hit, you have to find a pathway to hit. And interestingly, underlying a common pathway, there is angiogenesis, underlies 10 of these 12 pathways that has been described. So again, makes angiogenesis, Judy Folkman's original idea, maybe an attractive target too, but again, a lot of research needs to be done. So translational research, basic science, clinical science, then it affected the drug discovery, treatment, the biomarkers. What it did unexpectedly is the translation science changed the clinical trial design in the old-fashioned way. Traditionally, historically, you have phase 1, phase 2, phase 3 randomized double-blind. Now, what's happening is with the targeted agents, clinical trial design is changing. They are getting larger or smaller SMART trials, targeted trials. And one of the early adopters of this I-SPY 2 group, they just presented in San Francisco, California group, they presented some data on this on recent AACR a couple of weeks ago. So they started the I-SPY 2, I-SPY design adaptive trials. Instead of getting one drug and randomizing and developing it, they get 5, 6 drugs, and they put it in drug 1, drug 2, drug 3, and it uses genetic profiles to highlight the biomarkers differences. Then they put them through as they proceed. They change the drugs and they eliminate the people who respond, who don't respond, and they come up with a drug that is really effective and targeted. With this at CHI, we are working on this with Novartis company to come up with the signature trial design. You get the purpose of the study to determine whether treatment with the study drug improves the clinical benefit rates in patients with selected tumors. So the tissue sample collected, treating physician takes it, sends it for microarray-based genomic analysis. Treating physician, the gene expression is reported. Eligible patients, then there are 6, 7 different drugs that hits those driver mutations or targets. And they are put on the study. It's in a way they call the P2P, protocol to patient. So you activate the protocol when you discover a mutation and you have the drug, and for every patient you open a clinical trial in a way. But it's much more efficacious way of getting drug. Then that's the clinical trial part. There's also reflection this into the just regular general practice. Some companies already in the way of doing this. One of them is here as shown. And there are techniques that whole genome sequencing, exome sequencing, and targeted sequencing. They give you a result like this. You get a tissue, you run out of all the options, patient is in good performance status, you want to still be able to do something, no clinical trials. So they test this tissue, and they describe the genetic abnormalities. They say there is FDA-approved drugs out there, doctor. These drugs are available, FDA-approved for that particular cancer, or there are drugs out there, but not approved for that particular cancer. Then they give you, I put some examples here. So if the physician, practicing physician, regardless being on a clinical trial or not, you got your tissue sent, I hope the reimbursement will catch up with it, then they report you, okay, you're, let's say, pancreatic cancer patient, there's nothing else you can do. We discovered a EGFR mutation, lapatinib or cetuximab may be likely to work, but it's not approved for pancreatic cancer. Then you can call, and if you get the reimbursement, you can just do it and see the response and record it. And I will come to that, the electronic records affecting it. To leave this topic, I show you this paper, very amusing and smart too. The Dr. Gordon Smith, professor, published this in British Medical Journal to show a message to the, you always want to randomize clinical trials, but was there any trial that showed the efficacy and the safety of parachutes? And he just got it published, believe it or not. The author says that the second author tried to make him stop this doing, and he pushed, and he put very good jokes, but he edited some, removed some of the best jokes on the paper. But it is published as parachute used to prevent that and major trauma related to gravitational challenge, systemic review of randomized clinical trials. They did do the search, and they did find no clinical trials showing the data. So the point is, you don't have to have a randomized clinical trial to show that crizotinib is better than chemotherapy. Poor people, they will be using all that drugs, and I get the warning that I will be going fast, I'm sorry. It is then, what I want to connect here, my last part is the, so the basic science, the clinical science, the drugs, the biomarkers, and the clinical trial design, yet one thing arose out of nowhere is the electronic medical records. And as painful, as bad as it is, which I'm going through it right now, it has a huge potential. When you, like the whole community, whole world becomes the lab. Because as the prior slide I showed, you could use the tissue test and all that to use it. When, imagine everything goes into the electronic medical system, so you have a precious data accumulating there to be analyzed future times. Yet, these guys from Oklahoma, they came up with this another very fascinating, intriguing concept. Iridescent stands for implicit relationship identification in silico construction. What they do is Medline search. There is like 12 million published records in Medline as of 2012, and every year there is 500,000 added to it. So what they came up with, an interesting computer design, they get a topic, let's say it's the type 2 diabetes or single nucleotide polymorphism in certain disease. They put that name, query, and then direct, indirect relationships of that original topic, indirect, direct relationships, indirect, they match up, the computer does that tedious work, and they come up unexpected, miraculous solutions. That one best example was, they found that when they analyzed the cardiac hypertrophy, chlorpromazine, nobody would even imagine that would have any effect on the hypertrophy. Turns out that it reduces the hypertrophy, and they tested backwards, and they find that it is correct. And then they did check for type 2 diabetes. They found some epigenetic mechanisms for type 2 diabetes. And then, so the point here is all that science joined with the biomedical, electronic medical records, it is creating a huge, huge potential for discovery. And then I see this all the time. Patients come to me with that. So finishing here, Who are we? CHI. As I said at the beginning, I am so impressed with CHI vision. It is amazing. I would not expect from, it's a corporate organization, but this is such a scientific thinking that biospecimen, Central for Translation Research, it is applaudable. So what we are, who we are, we have 80 plus hospitals across the United States, and then I believe 17 of them are oncology cancer centers. And then we have been part of the NCCC. But among these cancer programs at CHI, we have 3 academic centers. We have several CCOPs, NCCC sites, pilot sites, and several rural hospitals, which one of mine is the rural hospital. And then how do we apply this great project, great vision into whole CHI across the United States? Clinical care of cancer patients, rapid changes, ethics, regulatory considerations, all that. How do we put these pieces together? So I tried to come up from my being in the trenches every day. Biobanking is a great first step, coordinated efforts, operational informatics support standards. Then genomic technologies, I believe Jeff Otherwise, his team will be doing a great job in putting pieces together. Then informatics, coming up here is our EHR status, National Oncology Service Line, Dax and his group, and they are doing a great job. So it is amazing. We just became Mosaic Electa. Here is the, you see the blue ones, the majority of our sites in cancer at CHI are Mosaic, and then there's some Epic and variant. The point here is that you have a central EHR, all of a sudden all the data is there, and you can do another lab sites like bench, bedside, and the EHR, you can search on it and come up with very meaningful solutions or discoveries. So then it will possibly in the future will get the biostatistics and decision making. So I'm finishing that. We had our first ASCO booth last year, and I wanted to share this picture. Again, great leadership, great support, great organization. CHI is amazing. That I'm more than impressed, and I want to thank you all listening, and also thank our little animal friends for getting into trials without consent, and then thank everybody else, and happy to answer questions. I rushed, I'm sorry.